Right, so on to gene expression in eukaryotes. So we're going to leave the prokaryotic world behind momentarily and look at the more complex situations in eukaryotes. So in this case, we have something that prokaryotes do not have. So let's take a prokaryotic cell and a eukaryotic cell and compare them side by side. So in the prokaryotic cell, we generally have one big bag of stuff, right? There's not really that much compartmentalization. We're going to exclude the inner membrane space for a moment. Just consider the cytoplasm as one big compartment. So let's do our processes we just talked about of making RNA, and then that RNA serving as a message to make protein. So in the prokaryotic cell, we have our RNA polymerase, which we talked about in the last lecture. Reads the DNA template strand. Again, I got my color coding here for you. The template strand is red, the coding strand is blue, and the new mRNA is green. As soon as it comes out the back of the RNA polymerase that you saw in the last lecture, there will be a ribosome there ready to grab it. Okay, so the ribosome, with its large number of RNA pieces and proteins, will grab this messenger RNA as soon as it comes out the back of the RNA polymerase. It's not even finished making this message, and it's already being threaded through a ribosome. So as it's being threaded through that first ribosome, it's making protein, shown here as the little blue spheres that are coming out the back of the ribosome. And then as it exits that ribosome, it enters another, which begins making the same protein. And as it exits that one, it enters another. And we can have a long string of these ribosomes. And of course, the farther down the chain you are, the closer to the RNA polymerase, the longer the protein you've completed at that point. So in prokaryotes, transcription and translation occur simultaneously. So translation begins before transcription has even completed. Okay? That is not true in eukaryotes. Okay? And what's the reason? So in eukaryotes, we do RNA transcription the exact same way. The polymerase is a little bigger and there's more proteins involved in finding the right spot, and generally the RNA is a little longer, but for the most part, it's made the same way. That mRNA is then exported from the nucleus to the cytoplasm, and then ribosomes can find it. So here, transcription must be complete before translation begins. That is not true of prokaryotes. So what's the advantages and disadvantages? For the prokaryote, this is so much faster. I can start making the protein long before the messenger RNA is even finished. And I can make several of them at the same time before I even finish the message. So this is speed. I can make lots of protein, the same protein, from one message as fast as possible. For the eukaryote, this is much slower. I must first complete transcription before translation can begin. However, there is an advantage. I can take that mRNA I just finished transcribing and do some processing. So I can cut out some pieces of it, put some other pieces back together, modify it, do a lot of things to it, which we'll talk about later, and then export it to the cytoplasm. So from one message, mRNA, from one pre-mRNA we call it, I can do all that processing and maybe make multiple proteins from it. Not the same protein, but different proteins. We're going to talk about splicing out introns and putting exons back together later. But I can make multiple different products from the same piece of original DNA, whereas prokaryotes can't do that. I make one message, it makes the same protein every time. So how does a prokaryote get around this? Well, prokaryotes generally make things called multi-cistronic messages. So one piece of mRNA encodes for many proteins, maybe 2, 3, 4 proteins. And if you look back at your lac operon, you'll see that was an example of that. So we have several proteins being encoded by the same message, and the ribosomes hop on immediately and start making all of them. Whereas in eukaryotes, we generally could make multiple proteins depending how we process the mRNA, but once we process it, you only make one, right? The final constructed mRNA generally only codes for one protein. So it's a 1-to-1 ratio there. But our diversity from the same piece of original DNA is much more robust than for prokaryotes. So what allows this difference between the two? What's the major difference between the prokaryote and the eukaryote here is the fact that we can isolate the nucleus. So it must be this nuclear membrane. Without the nuclear membrane, the ribosomes would have access to this message the moment it starts being made, like a prokaryote. But in eukaryotes, the ribosomes, once they're folded up, can't get back in the nucleus. Remember, they're made of RNA, so they're made there as well. They're exported, and they get folded up into the ribosome, the rRNA, and they can't get back in the nucleus. They're just too big to fit through the pores. So it's kind of a sequestered region where I can do all this RNA processing, then send it out to the cytoplasm. Okay, so let's look at our different RNA polymerases and see what they do, because it's not just one. We have several of them. We're going to talk about 3 types. Okay, so we have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And they make different things. They do all make RNA, that's true, but they make different RNAs. So let's read them off to the list here and see how we can categorize them and distinguish them and maybe discover which was created or invented first. So type 1 is found in the nucleus, like all of them are, but in a specialized part of the nucleus called the nucleolus. Okay, so we're clearly talking about eukaryotic RNA polymerases here. So type 1s make mostly rRNAs. So they're the ones responsible for making all the rRNAs. Remember, this is 80% of the RNA in a cell. So they're the workhorses of making RNA. They don't make any of the other types. They don't make mRNA and small nuclear RNAs or stuff like that. They make rRNA, and they make one tRNA, just one. And we'll get back to that foreign exchange student in a minute. So type 1 eukaryotic rRNA polymerases, or RNA polymerases, make rRNA. Okay? Type 2s make mRNAs and the small RNAs, right? Like the small nuclear RNAs. And then type 3s make all the tRNAs except one, and they make one of the rRNAs. Okay, so if you think about it, let's talk about numbers 1 and 3 together. Numbers 1 and 3 make all of the rRNAs and all the tRNAs, except they have this foreign exchange program going on, right? Type 1 makes all the rRNA except the smallest one. You notice the 5S? What was that? So the type 1 makes all the rRNAs except the smallest one, which is made by type 3. And type 3 makes all of the tRNAs except one, which is embedded in the middle of the rRNAs that type 1 makes. So they have a foreign exchange program going on. So type 1 makes all of one type except for one of them, and type 3 makes the other one. And type 3 makes all of the tRNAs except for one, which was made by type 1. So they've kind of exchanged partners there. They require each other to make all those parts. Type 2 is a little different. It makes only one thing to itself, no other parts needed. It makes all the mRNAs and the associated small nuclear RNAs, which those things are used to splice the mRNAs, which we'll talk about later as well. So if we look at these and look at their effects from this little toxin made by this mushroom, it's called alpha-amanitin, and it's the picture shown at the top right, the mushroom that makes it. So you don't want to eat this mushroom. And this it looks like a protein, but it's highly modified protein. It's cyclic, and it's got some branching going on and other oxidation events. But this little protein, if you add it to solutions of these RNA polymerases, it will strongly inhibit type 2. It will shut down RNA polymerase type 2. It will not function in the presence of this molecule. However, type 1s are insensitive to it. You can add as much as you want. It will not inhibit RNA polymerase type 1. So there's clearly a difference in them. And type 3s will only be inhibited if you add this in high concentration. So it's somewhat sensitive to it, right? It's not strongly inhibited, and it's not totally insensitive, but somewhat sensitive to it. So if we rank those, this mushroom is making this toxin for what reason? Well, not so much that it's going to eat other things. It's a fungus, right? But it's competing with bacteria and other fungi and other organisms for the same food. So if it makes this toxin and it inhibits RNA polymerase in general, it would kill itself. That's not good. So if it makes this toxin and it inhibits its competition's RNA polymerase, but not its own, that's a perfect weapon, right? So it's inhibiting its competition's RNA polymerase type 2, but not its own. That tells me RNA polymerases type 2s from different organisms are diverse. They're different from one another. Whereas type 1s, no matter how much of this I add, it will not inhibit it. So the mushroom, not necessarily has figured it out. I'm giving it anthropomorphic qualities now. But the mushroom has evolutionarily learned don't inhibit type 1 because all of ours are the same. So type 3 is somewhere in between. So this gives us some evidence that type 1 RNA polymerase, which makes the most important pieces of RNA, the ribosomes themselves, is the same universally across all species, and that's true. So it's the most ancient of the RNA polymerases. Whereas the other two are more recent evolutionarily. tRNAs and mRNAs especially are quite different among different species because they recognize different proteins, right? They recognize different precursors. So type 1 must be the oldest evolutionarily, followed by type 3, and then a more recent, the diversity of the type 2s. So let's look at those three polymerases one more time, and not necessarily in the order on the screen here. Let's start with the middle one, RNA polymerase type 1, the most ancient of the bunch. And I'd like to know, if I'm RNA polymerase type 1, where should I start making my RNA? Remember, I'm going to approach a DNA template somewhere. There's going to be a TA binding protein that's going to tell me to bind in a certain spot. And then I need more instructions because that's all over the place. Which one of those proteins should I make? I'm looking to make rRNAs, not necessarily proteins. So I need some direction other than just a TA binding protein. So all the RNA polymerase type 1s use elements in the DNA that are upstream of the start site, or 5' to the start site. What that means is, on the diagram, if you look at this diagram, this is a piece of DNA serving as the template, the coding and template strands, and the arrow is where we're going to start making the RNA, that little up and over arrow. The promoter region is where some proteins bind to tell RNA polymerase type 1 start here. And the information that tells it where to start is always just to the left or upstream of the start site. It's always 5' to it. It's always upstream every time. This is the most ancient way to do things. Okay? Move on to RNA polymerase type 3. Right? This one makes all the tRNAs except for one, and the smallest piece of our RNA, the one that type 1 didn't make. Okay? So RNA polymerase type 3, everything that tells it where to start, it figured out a new strategy. Everything is downstream of the start site. Okay? Every single piece of information that says start here, the information on that is downstream from it. So completely opposite from type 1, or from polymerase 1. Unfortunately, these were named RNA polymerase 3 type 1 promoter and RNA polymerase 3 type 2, which is insanely confusing. So instead, I just want you to remember RNA polymerase 1 always uses upstream promoter elements. You don't have to memorize all the alphabet soup of acronyms here. So type 1, RNA polymerase type 1, uses information that is upstream of where it starts. RNA polymerase 3, a little more recent evolutionarily, has a new method, everything downstream of it. And then the most recent evolutionarily, at the top, RNA polymerase type 2 says, I'll take the best of both worlds. Sometimes I'll use things upstream, sometimes I'll use things downstream, and sometimes I'll use both. So it's the most recent and takes advantage of all those opportunities. So evolutionarily, this also makes sense. Type 1, everything upstream tells it where to bind and where to start. Type 3, polymerase type 3, everything is downstream of the start site, nothing upstream. And type 2 uses a combination of one or the other or both. So what are these little recognition sites? What are we talking about here? So the first one we talked about was this TATA box. And what these numbers on the top mean here, so we're going to use RNA polymerase type 2 as our example here. It's looking for our TATA box, right? And when a protein finds it, it has to recognize TATA on that strand, and on the other strand underneath would read 5 to 3, TATA underneath it. So the numbers you see here next to these letters aren't the numbers in the sequence, because they clearly aren't sequential, but they represent the percentage of the time you're going to find that particular base there. And you see the AT in the middle is almost universal, 97 and 93% of the time. And the others aren't far from that. They're very high percentages. So for the most part, we're going to find TA every time, right? There's very few that aren't that. Okay, so there's a protein that looks for that. There's some other ones. So if you look back at our chart here, we had some boxes that said enhancer in blue and yellow UPEs and downstream elements in black box, C and B and A at the bottom and so forth. Don't worry about all the acronyms. Just know that some of these things are like the TATA box where they have a certain sequence, but it's not TATA. We have one called a CAT box, and it's not just CAAT. It's got to be in context. So there must be something outside of that as well. You see the generalized context here. The one at the bottom is called a GC box, and of course G followed by C happens all over the place, but it must be surrounded by a pair of G's on each side. It's just called the GC box. So the sequence doesn't occur that often, and a protein can find it. So if a protein were to find the right elements, RNA polymerase 1, 2, or 3 can show up and say, this is where I'm supposed to start. Now, when do you find that element? There's going to be a protein involved. So a particular protein might be looking for that GC box, and RNA polymerase 2 might recognize that protein once it's bound. But if I don't make that protein, I'm not going to make that particular mRNA. So I have control over things. And this is an example of one of those proteins here. This is the protein that finds the TATA region, right? And we give names to these things like TATA box binding protein. We also give them more standardized names like TF2D, right? So TF stands for transcription factor because it's involved in transcription. 2 means it works for RNA polymerase type 2. And D means it's the fourth one that was given a name in this sequence. So there's TF2A, TF2B, TF2C, etc. So we just keep naming them for that. This one happened to get TF2D as a name in my example. Okay? So look at the bottom. This is the picture here, the TATA box binding protein. What does it do? Well, it recognizes the TATA on one strand and the appropriate TATA going the other direction on the other strand, and it acts like a little saddle. It binds on the DNA. And if I had to ask you, or if I asked you, what part of the DNA would this thing recognize? And your answer choices might be it recognizes the backbone or it recognizes somewhere in a groove. Which one would you choose?
Somewhere in a groove.
Why would you think groove? It's right, but why would you say that?
Some specific sequence that the enzyme would recognize or protein would recognize.
So the protein needs to recognize a specific sequence, absolutely. Why can't it do that by binding to the backbone?
All the backbones are the same.
Exactly. The backbone is the same, in this case, deoxyribose phosphate everywhere. Now there may be slight differences in the way the backbone is shaped because of the base pairings, maybe a high GC rich region, for instance. But I'm looking for a specific 4-base sequence, and I need to get in the groove there to figure that out. So in the groove, you have access to the base pairs, and I can tell if it's an AT or a TA or a GC or a CG pair. And that's what this protein does. And it recognizes those 4 bases, the TATA, and in the other strand, TATA. Again, palindromic, that's the general theme for these things. And what it does is it binds on it, hops on it like a little saddle there, and it starts to unwind the DNA and bend it a little. If you notice, you look at the blue strand there for the backbone, it's not a nice curve anymore. It's got a bend in it. So it distorts the DNA a little bit. It's not really acting as a helicase. It's not unwinding it completely from the other strand because then it won't recognize it anymore. It's looking for a groove, remember. But it distorts it enough that it looks different here now. And other proteins will do this in their areas, and RNA polymerase, in this case type 2, will come along, recognize these proteins, recognize the bent DNA, and go, thank you, I will start here. That's how all this works. Okay? It's not the only one around. So here we have at the bottom, we have a transcription factor shown in the red color, RNA polymerase in green, and then we have a bunch of blue proteins here called mediator, right, or mediators. And I don't really want you to get bogged down in the nomenclature too much, but if we call something a transcription factor, it could have one of two meanings. It could mean it's involved in transcription. That's what I'm going to use it for because that name makes sense. Or it could be a protein involved in transcription that specifically interacts with DNA. And those that do not interact with the DNA, while we'll still think of them as helpers in transcription, sometimes don't get the moniker transcription factor, and we just call them a mediator. And they mediate or bridge the interaction between a true transcription factor and an RNA polymerase. But I'm not going to ask you any of those specific details. A transcription factor, what I want you to know, is involved in transcription. It's one of these things that helps the RNA polymerase or hinders the RNA polymerase from doing its job. If it helps it, it's a positive thing, right? It's a positive transcription factor, like an enhancer might be a name for one of those things. If it hinders it, we might call it a regulatory molecule or a detractor or an inhibitor or something like that. So they can either help or inhibit the process. And that gives me control over which proteins or which RNAs I'm going to make. If one cell, you can imagine it like the old NASA control rooms when they were doing the Apollo missions, right? There's 50 people sitting at computers and one guy asking, are we good to go? And I got to get 50 yes answers for us to do something. That's kind of what RNA polymerase is doing. It's sitting here waiting for, say, 50 molecules like transcription factors and mediators or whatever to get their act together, find their spot, and tell me I'm good to go. The cell can simply don't make one of them. Leave one of the mediators out. I was told not to make that protein. Therefore, we don't go. We don't make this other RNA. So we have lots of places we can control this. And there's lots of overlap and crosstalk to control these proteins. Okay? So let's do an example of one of these, right? A very specific example. So we're going to use a molecule like estradiol here, shown on the top right, and it's going to be our signal molecule, right? So this is a molecule that one cell is making and secreting or releasing into the environment, and it's going to tell another cell to do something. So it's cell-to-cell communication. So in this case, the estradiol it's a steroid-derived hormone. It's being made by the cells in the ovary and released into the body. So the ovarian cells are telling many, many other cells that are not next to them what to do, right? So it's long-distance communication using this smoke signal of a steroid molecule. So number one, this molecule is not soluble in water, right? So it's a lipid, right? It's based on cholesterol. It's made from cholesterol. So we need to carry it to its new destination. So there'll be some steroid-carrying protein in the blood that carries this like an albumin to its new home. And its new home being wherever it can find a receptor. So what happens is this is released by the ovaries. It's bound by proteins, taxis, that carry it all throughout the bloodstream. And it's released to virtually every cell in the body. Being a steroid, a lipid-based molecule, it has no issue getting through the membrane, right? Because the membrane is a lipid bilayer. So this thing goes right through the membrane. However, we need to get it to the nucleus so it can act as a transcription factor. But from the estradiol's point of view, the cytoplasm is an ocean that I can't cross because that's water environment. I was dropped off at the cell surface. I've made my way inside rather easily, but I can't get to the nucleus. I can't cross this ocean of cytoplasm. So I need another carrier. So the receptor for this hormone is not on the outside of the cell like many receptors are. This one's found on the inside of the cell. It's bound to the inner leaflet of the cell membrane because that's how far this molecule can get on its own. So that molecule on the inside, the steroid receptor, which is convenient because you keep it in your cell all the time, you don't have to export it, grabs the estradiol or estrogen molecule and leaves the cell surface. Now, it is a soluble receptor. It grabs this molecule and floats over to the nucleus. It can go through the pores of the nuclear membrane, no issue, and it finds a piece of DNA. These steroid receptors have parts of them that bind to DNA and a part of them that bind to the ligand here, right, the estradiol molecule. So when it binds to its ligand and it goes and binds to the DNA, it's acting as a triggered transcription factor. If it doesn't bind its ligand estradiol, it won't act as a transcription factor. So one cell, by releasing this, tells another cell to make a functional transcription factor and make this other RNA. So it's telling another RNA what to do, another cell what to do. And here's a picture of that process here. Shown in the linear diagram is the straight chain of this protein, and certain parts of it perform certain functions. It has a DNA binding domain with a bunch of zinc fingers, they're called. It's 4 cysteine residues, and I know you're learning your amino acids, so the cysteine has a sulfhydryl group on the end of it, so it's an SH. It loses the H, and then the 4 sulfurs bind to this +2 zinc, right, the zinc ion. And the reason it does that is causes the protein to adopt a certain shape. It's just an architectural thing. And because it adopts that certain shape, you notice these alpha helices, right? There's 4 of them here at the top, are placed in the grooves of the DNA. So this makes a great DNA binding pocket. Okay? Another part of the molecule, the part that binds the ligand, in this case our estradiol, has a hydrophobic pocket as part of itself. And since the steroid is hydrophobic, it'll bind in there next to all those hydrophobic amino acids that line this pocket. Okay? These are part of the same protein, but this pocket's where the steroid binds, and then the other end of the molecule is where it interacts with the DNA. Okay, so let's answer some of these questions on here. I said this thing has 9 conserved cysteine residues, 8 of which are involved in these zinc fingers. So what does that tell you about the geometry coordinating a zinc ion? If it takes 4 cysteines to coordinate the zinc, what is its geometry?
Tetrahedral?
It's tetrahedral. And if you look at the picture above, you see that it might be a little small, but it's definitely tetrahedral. If you're in the lab course, we're going to look at a lot of these structures later towards the end of the course, and you'll see that it really is tetrahedral as we look at it in 3 dimensions. So on the right, I have another question. The ligand binding pocket is hydrophobic. Why is it hydrophobic? So why is our ligand binding pocket lined with hydrophobic amino acids like leucines and lysine?
So,
go ahead.
I was going to say because so it could interact with the hydrophobic steroid.
Right. It's very specific about what hormone it interacts with. So if it's estradiol or testosterone or progesterone, not only is it hydrophobic, but it has to be specifically shaped. Right. So this thing is definitely hydrophobic to eliminate any water-soluble things from getting in there, at least any big water-soluble things. And it has to match the shape of the ligand it's looking for. So they're very specific for certain things. All right. Once this thing binds to it, I have a little process here at the top, shows you what happens. So this protein showing you the ligand binding region, when the estradiol binds to it, there's a little helix on the outside called helix 12, because it's the 12th helix in number in the sequence. It is more stable if it flips over to the side, right? It kind of latches onto the side of the protein. So instead of pointing out in the front, it latches onto the side. And of course, it makes the protein have a different overall shape, right? So only if estradiol binds will it do that. So in the cartoon form below, we have our receptor sitting there with its little purple helix sticking out and the ligand site empty. Of course, the other end of the molecule could bind to DNA, but even if it does bind to the DNA, the coactivator or mediator or other transcription factor or RNA polymerase, whatever it binds to, usually some coactivator we call it, usually a mediator, doesn't recognize it unless the helix flips over. And it'll only do that if this thing has migrated out to the cell surface, picked up an estradiol molecule, because that's the only place it can find them, come back to the nucleus, bind in a pairing like this, in a dimer, right? And then the coactivator will find it. And once it does, we now have a signal that says, let's make this protein. So this thing is one of these activators that you see in these big red blobs here, or on the left figure, it's one of the bluer molecules with its hormone attached. And many, many other, what they call transcription factors here. It looks like a yellow paw print on the left. We could call those mediators if you wanted to. And there's maybe one bound to the TATA promoter called the TATA binding protein down there in purple. All those things come together to tell RNA polymerase II, this is the right place to start making this protein. Start making this RNA, sorry. Right? We could also have things called silencers and repressors. So you see the top figure, there's this repressor which binds to a silencer, and when it does, it prevents some other activator from binding. Not shown in this picture, of course, this is the activating complex. But if that repressor were to bind, perhaps one of those big red activators could not bind, and that's just as effective as not having it there. So I could have another protein being made by the cell that says, yes, I know we received an estradiol signal, we should be making this RNA, but I'm vetoing that action because we're not that kind of cell. So different cells can respond differently to the same signal. Okay? And then the last thing I want to cover is how do we control all this, right? So the first thing we need to do in order to have all this stuff bind to our DNA is our DNA must be accessible. And you told me, what do these transcription factors look for? What part of the DNA are they binding to? And you said, accurately, they're going to bind in the grooves. They're not necessarily looking for the backbone, because that's the same everywhere. We're going to look for the grooves for the most part. To have access to the grooves, this thing needs to be open and accessible. And for the most part, we have it wrapped up around histones. Remember all those proteins that we have our DNA wrapped around to facilitate compacting the DNA as storage. At some point, I want to remove a piece of it from a histone. I don't want to undo all of it because it won't fit in the cell at that point. But I want to undo some of it, open a local region, and make the gene product that's on that region. So what happens is I need to get the DNA off of the histone. And the histones have what charge? The DNA is negatively charged all across its backbone. Our histone proteins tend to have what charges on their surface?
Positive.
Positive. I know you're learning your amino acids. What amino acids at pH 7 might have a positive charge? So pH 7 would be considered acidic compared to some pKa's, right? If it's a smaller number. So you're looking for proteins or amino acids with pKa's above 7. Which ones are those? I can think of two.
Lysine and arginine.
Lysine and arginine, your basic amino acids, right? Not histidine, even though we call it a basic one, its pKa is more around 6. So it would not be protonated in this case. So the pKa being larger than the pH, it's mainly going to be protonated. So we have the lysines and arginines on these histones. Let's focus on the lysine example. So at the top I have a backbone protein chain sitting there with a lysine R group off the top of it, and it's protonated. Because it's protonated, it has a positive charge, and the DNA will like to stick to this type of protein. How can I get the DNA off of that protein? Well, I don't want to destroy the protein. I'd like to use it again at some point. I just want to mask the positive charge. So I don't want to cut off the amino group. I want to just mask it temporarily. So I'll use this molecule called acetyl-CoA. And the CoA part, think of it as just the handler for the acetyl group, which is the important part. So the CoA is just the carrier, the taxi. And I'm going to put the acetate group, the latter two carbons you see there, on the sulfur, from the sulfur, and transfer it onto the nitrogen of that amino group. Okay, and of course the CoA leaves. When I put it on there, it'll now be what type of arrangement up here? What kind of bond is this? When I attach this acetate to an amino group.
Amide.
It's an amide bond, right? It's not a peptide bond because this is not making a amino acid connection between two amino acids. It's just an amide bond. And it's fairly stable, right? This is a very stable type of bond. It won't fall apart readily. But it also masks our positive charge on the nitrogen. So this looks uncharged now to our DNA. So if I start acetylating a lot of these histones with their lysine tails, it won't stick to the DNA as tightly, and things like topoisomerases and helicases can easily get access just by nudging the histones out of the way, right? So I can open up the DNA and make it accessible to RNA polymerase or transcription factors or whatever else I need to. The enzyme that puts this acetyl group on, it's transferring it off of CoA and onto the lysine, is called an acetyltransferase, right? It's putting it on a histone, so it's a histone acetyltransferase, abbreviated HATS. Okay? Do I want this situation to remain like this permanently? No. Maybe I don't want to make this gene anymore. I got a different signal. I'm told to make something else. Let's shut this process down. So when I'm done making this, maybe the transcription factors aren't showing up anymore, no RNA polymerase around. I need to package the DNA up and put it away for now. So those are going to be removing these acetate groups. And that's just going to be a hydrolase. Right? It's going to come along and break this bond using water. And it cuts off this, making acetate, and leaving the amino group behind, which will immediately get protonated because it's in an environment that's more acidic than its pKa. And so we'll be back to a charged lysine. And the DNA will readily bind to this again. Okay? Those are called histone deacetylases, or HDACs. Okay? What does this accomplish? It puts the DNA away. Right? The first one accomplished making the DNA accessible for transcription. The latter one puts it away so we don't transcribe it and we don't need it out right now. A few years ago, I had a group of students come up, I had them make up their own analogies, and this one was spectacular. I'll make it PG-13 because theirs was not, but it was hilarious when they gave it in class. So they described the hats and the HDACs as people. So let's say you go to the library and you're in a time where you have to get help from the librarian to go find a book. I know you just look it up on the internet today, but let's go back in time to 1980. So when I was going to go to the library and look up a book. So you go and look up a book, you don't know how to use the card catalog, it doesn't make sense, you need help from a librarian, right? So there's this nice, young, helpful librarian. She's still new, so she's not so mean to you. So she comes over and she's very helpful and she helps you find the book you're looking for, right? Takes you up to the 3rd floor of the library, whatever it is, says it's on this shelf, pulls the book out for you. Here you go, read what you want. And what do all librarians and all libraries tell you to do with the book when you're done? Don't put it back because you're going to do it wrong. Just leave it on the counter, leave it on the bench, leave it on the table. We'll put it up ourselves because they want to put it back correctly. They don't trust us. So this is the histone acetyltransferase, the young, helpful librarian, the new librarian who's willing to help you find what you're looking for. So go searching along the DNA, remove the histones out of your way, open the book, here, read what you want to read. Okay? And then later on, you just leave the book there. And of course, you do this all over the cell. We have lots of DNA opened up to make lots of proteins, and everybody just leaves. Then there's the old, less helpful, probably more grumpy, eyeglasses on a chain librarian who's been there for 50 years. She doesn't talk to you very much. She just grumpily puts all the books back up. That's Mrs. HDAC at the bottom. Right, so she comes along and sees all this DNA and histones that are not put together, removes all these, acetate groups off the lysines and wraps it up around the DNA again. Okay. We won't get into the other parts of their analogy where Mrs. HDAC had a night job at a different establishment, but that's a lecture for another day. Okay, so let's put it all together in a figure here. So to start off, if you want to make a piece of RNA, right, which might be used to code for a protein, the first thing we need to do is unwind our DNA. So the transcription factor will find a spot, right? It binds, perhaps it's our estradiol one. Then some coactivators bind, mediators, other transcription factors, and realize, hey, our DNA is still wrapped up around these histones. I need access. So they show them their library card, and the hats come along and remove all those DNAs from the histones by putting the acetate groups on. The remodeling engine helps to accomplish that. It's just somebody that comes along and grabs the book for you and opens it for you. You have access to a piece of DNA. Here are the exposed site. Maybe dozens of other transcription factors come along and say, yes, this is correct. Or none of the negative ones say stop. And then an RNA polymerase is brought along with mediators and many other transcription factors and says, yes, we're making this RNA, let's do it. So we start making the RNA. We continue until either we run into a row-dependent or row-independent termination, and eventually the RNA is pulled away, the RNA polymerase falls apart, goes away, and we have this exposed site again. Mrs. HDAC comes along, removes all those acetates on the lysine tails, the histone wraps around the DNA again, or the DNA wraps around the histone again, and we're back where we started, unless another transcription factor says, hey, we need to make more of this, let's continue. Or we're told to stop doing it. And this is the whole process, and this goes along continuously in the cells, and they're constantly receiving signals that talk to each other. So this is very complex inside the cell, but this is essentially what a cell does all the time. Okay? So our next lecture, we'll talk about what happens to the RNA that is released at the end of that, and in prokaryotes, virtually nothing happens to it. There's a ribosome already on it, probably several already on it before we finished. However, in eukaryotes, we do a lot of processing, and we'll talk about that next time.